Hello students, welcome to this session Hematology basic requirement. So now first, what is hematology? Hematology is study of blood. Hem is blood and logy is study. So today we are going to study what are the things required to start the hematology laboratory and investigations. So first thing required in hematology laboratory is hemoglobinometer. So hemoglobinometer is also called as Siley's apparatus. It consists of hemoglobin pipette, hemoglobin tube and comparator to compare the readings with the standard glass. And the uses of the hemoglobinometer is hemoglobin estimation, and depending on that we can divide the anemia, whether it is mild, moderate or severe. Second thing required for the hematology laboratory is RBC pipette. So this pipette has a large bulb which contains red glass bead, so that we can differentiate it from the WBC pipette which contains white glass bead. It has three markings: 0.5, 1 and 101. So blood we have to take up to 0.5 marking, and then diluting fluid up to 101 marking, so that the dilution of blood with the diluting fluid is 1:200 for doing RBC count. So uses of RBC pipette is one, RBC count estimation, and second, platelet count estimation. Third thing is WBC pipette. It is smaller than red seed pipette, and its bulb is also smaller, which contains white bead. It has three markings: 0.5, 1 and 11. So blood we have to take up to 0.5 marking, and WBC diluting fluid up to 11 marking, so that dilution is 1:20. So uses of the WBC pipette are to count total WBC in blood, to count total WBC in all body fluids like CSF, peritoneal fluid, pleural fluid, pericardial fluid, synovial fluid, and also total sperm count in semen. Fourth thing is newborn's chamber. It is also called as counting chamber. So it has area of 9 mm and depth of 0.1 mm. It has two stages separated by a gutter, that is a space. So 9 square millimeter area of counting chamber is divided into 9 squares, that is 1 square is 1 square millimeter equally. The four corner squares measure 1 square millimeter and divided into 16 small squares and are used for counting WBCs. Central square is divided into 25 squares, and each one is again divided into 16 small squares, so that the central square is used for the counting of the red cells and platelets. The uses of the newborn's chamber to count WBC count, RBC count, platelet count, sperm count, and total cells in CSF, pleural fluid, ascitic fluid, synovial fluid, pericardial fluid. Fifth thing is Westergreen's tube. It is also called as ESR tube. It is 300 mm long, and graduations are 0 to 200 markings, and the uses is determination of the ESR, that is erythrocyte sedimentation rate. Sixth is the Ventrobes or hematocrit tube, also called as PCV tube. It is 110 mm long, and it has 3 mm internal diameter, and the markings are 0 to 10 and 10 to 0. So uses are the scale 0 to 10 used for ESR, and the scale 10 to 0 used for PCV, that is packed cell volume. These are my references from where I have taken my notes, and I hope you find this session very useful for your academics and for your future practice. Thank you so much.